Okay. So when I see movies like Harry Potter or any science fiction, one thing that truly excites me is when I see people going invisible. And that's why I thought of talking about this today. Now there's one point about watching a movie and being excited about it, but there's another point where we need to ask the question, is what we see, is something like that truly possible? So in this talk today, actually, I'll try to address the question, is invisibility, or can invisibility ever be a possibility? And before I start getting into the details of what's possible and what's not, let's first try to answer the question, what we need to make things look invisible. Okay? So this is a picture of a semi-invisible girl. The reason why I say it's semi-invisible is because you can see her body, but her head, hands, and legs seem invisible. Now if you think about it carefully, you'll realize the reason why her head seems invisible is actually because you can see what's behind her head, the mountains and the water. Or in other words, you can see through it. Okay? So this brings to our first point, is that if we want to make something invisible, we need to be able to see what's behind that object. Now what if we have a situation like this? Now in this case, the shoes, just imagine there's someone there, okay, and you can see through, but you do see a shadow. So if you have a shadow of an object, you cannot make it invisible. So that is the second point, is that you have to make the object lose its shadow. So these are the two main key things. Now, people have tried many different things to try to kind of see through things. And one of the most common and oldest thing is actually known as camouflage. So this picture is of a Chinese artist. His name is Liu Bolin. And you can see that if you look from the front, that he almost seems invisible except his head. Right? He uses this technique called camouflage, and it's actually a very simple thing. The idea of camouflage is that you basically color yourself to look like the background. For example, here, he first wears a green jacket and trouser. He colors partially himself up just to look like the stairs, and eventually he colors completely himself up just to look like the background. So if you see from the front, it kind of gives you an illusion that you can see what's behind it, which is the background, and that makes you think that he's invisible. But you have to remember it's not truly invisible, because for example, if you see these red lines here, you cannot see it here, and that's because he's actually obstructing the view what's behind him. And camouflage is actually something very common. If you look around in nature, for example, if you see a chameleon, you will see that a lot of animals actually change their color to kind of look like their background. Okay? So then there's another technique which is known as the retroreflective projection. The idea is to create semi-transparent cloaks like this. And this actually technique is again a very simple process. So what it does is that let's assume this is a person that we want to make invisible. So we put a camera behind this person. The camera takes an image of the background. The image is fed into a computer. The computer is connected to a projector, and the projector projects the image of the background into this person's cloak. So idea is that the cloak basically acts like a projecting screen like this. So because you see the image of the background, you think that you can kind of see through this person. So this kind of gives you the illusion as if the person is transparent. But you have to remember, just like this screen has a shadow, so will this person, and actually you can see it. So it is again not truly invisible. So to make something truly invisible, we really have to actually go into the physics of it. So let's first try to understand what is light. Okay? So light is actually a part of the family of waves, which we call as the electromagnetic waves. Now that includes a lot of waves. It includes the radio waves that we use for communication. It includes microwave that we use every day for cooking, and many different waves, all the way to X-ray that we use for medical applications. Okay? So the only thing that's different in all these waves is what we call as the wavelength. So what's a wavelength? Just like if you see a water wave, you see a high point, low point, high point, right? So the wavelength is the distance between these two high points. So when you compare a radio wave and an X-ray, the only thing that's changing is the distance between them. So an X-ray has a much shorter wavelength, which means you can imagine like you're squeezing the wave, or you're bringing these two peaks together. Now when we refer to light, the only part that we usually refer to of the part of the electromagnetic spectrum is this one. And just to give you an idea of what the wavelength sizes are, if you look at the radio wave, usually the wavelength is of the order of buildings that you see. The microwave that you use for cooking, for example, are usually orders of centimeters. So for example, a butterfly. Now the visible light that our eyes see, the wavelength is something comparable to the width of your hair, actually much smaller. Okay? So we now know what light is. Now let's see how we can manipulate light to make something invisible. Now we know that when you have a normal opaque object, it casts a shadow. The reason that is because light rays come, it hits the object, either it gets absorbed or it gets reflected. Now because these light rays cannot pass through this object, that's the reason why you cannot see through it, and also that's the reason why it casts a shadow. Now let's say we want to make this thing invisible. So what we have to do is that we have to take this object, we have to cover it with a cloak. And the cloak should be such that when the light rays hit it, it does not get reflected. But what happens, it goes around this object and it comes out just as there's nothing there, okay? So if you can make a cloak such that it can bend the light rays as if there was nothing there, so it just passes through, you can make this object lose its shadow and also see through, or in other words, you can make it invisible. Now the first question that should come to your mind now is, can we actually bend light? And the answer is yes. Light bends all the time. For example, if there is a beam of light, it's coming from air, goes to glass and then air, it bends. So as light propagates or travels from one medium or one material to another, it bends. And one example that you see in your everyday life is actually if you put a spoon in a glass of water which is half filled, if you look from the side, the spoon actually looks as if it's broken. And that's really because light bends as it goes from air to water. Now why does that happen? The reason it happens is because different materials or different medium, as we call, have a property which is different and that we call is a refractive index. We usually assign a number to the refractive index. For example, for water, it's 1.33. For glass, it's 1.5. And for diamond, for example, it's very high. It's 2.42. What this number mean is that higher this number is, more the light will bend as it comes from air. And actually, some of the amazing properties that you see for diamond, for example, the glitter and things like that, is actually because of its high refractive index. The refractive index is so high that light, when it comes from air, it bends so much, it kind of stays inside the diamond. And that's why you see the glitter. So now coming back to cloaking, right? So I said, if you want to make something invisible, you need to be able to bend light. We know that light can bend if you change the refractive index. And now we know that different materials have different refractive index. Now can we go ahead and make one cloak? Well, the problem is life is not that simple. The refractive index that I talked about in natural materials is actually a result or is due to the atoms and molecules of the material. Okay? So different materials have different atoms and molecules, or they have different chemistry. And if you want to change the refractive index of a natural material, you have to really change the atoms and molecules, and that's really difficult. Especially for cloaking, because you need such complex bending, so you need a very complex refractive index profile. And this actually was a big hindrance for a long time. The real breakthrough came when actually some scientists realized or actually found out that you don't really need to change atoms and molecules if you just want to change the refractive index. What instead you can do, you can take a natural material and you can build some units and you can stack them together. And by changing the shape and the way you stack, you can get any refractive index profile you want. Okay? However, there's one caveat, as these units, they need to be much smaller than the wavelength. For example, if you want to do an experiment in microwave, the wavelength of microwave is usually about some centimeters, so these units need to be, let's say, millimeters. But if you want to do something like invisible, because again the wavelength is so short, you need to make these units of the order of nanometers. Okay? So this is a typical material that I was talking about, the man-made material. Such materials are called metamaterials. The reason they're called meta is because in Greek, meta means going beyond nature. Okay? So you can see that these units actually are made of copper, so that's naturally available. But what they have done is that they've made these units of certain shape, they've stacked them together. And because they're stacked in this way, and based on how you stack it and what shape you have, you can have different properties. For example, here they wanted a different property, so the shape is different and they have stacked it differently. So these units actually, they were designed for radar signals. So each unit, and radar signal, the wavelength is usually about a few centimeters. So these units were actually in order of millimeters. So now, after this, this field of research came in, people were very excited that now we can design refractive index any way we want. And that actually led to the first demonstration of cloaking. Again, this cloaking first demonstration was done in microwaves. And the reason is because the metamaterials that were developed at that time was in the microwave regime. Because again, if you think about it, microwave has a much longer wavelength compared to visible light. So it's much easier to kind of make those units to tailor your refractive index. So this is the setup they had. The idea was, first, so this red, blue that you see is actually microwave. So they will shoot microwave, and this green box circle that you see actually is the cloak. So first they will remove the cloak, they will put an opaque object, copper cylinder actually. Then they will shoot microwave, then with an antenna they will detect where the microwave goes. So if it's an opaque object, like you expect, it will cast a shadow and it will reflect. Next, what they will do is that they'll keep the cylinder and they'll put the cloak back. And as I showed earlier, right, they will expect this microwave to come and not hit this cylinder here, but instead kind of bend around and go. So the metamaterial that they used to make this cloak look something like this. So this is they used, and this actually is the experimental data they have. So this, just a second. So the first case here is just a copper cylinder. Again, this red-blue that you see is again the microwave radiation. So you can see that it goes, and you see there's a huge color difference between what you see here and here. And that's because the light is actually, the microwave is actually not passing through this cylinder and is casting a shadow. Okay. So next what they will do is that they will put the cloak. Okay, and these lines here, they just show how the light travels. So if you see, what you will see is that, now you see the microwave radiation goes. Nothing goes inside here. It actually bends across here and then it leaves. Right? So this actually was the first demonstration and a proof of principle that cloaking actually is possible. Again, because this was the first experiment, it was not perfect. So you can see that there's a slight color difference here. It is a little lighter than here. In an ideal cloak, you should not have it. But that's, you can think is because it's not perfect, you have some sort of reflection. That's why it is casting a slight shadow. But again, it shows that in principle it's possible, right? So now that I have shown you that cloaking, the principle is possible, now the big question is, what is the future? Right? Can we actually make a Harry Potter cloak? Well, the unfortunate answer is no. And the reason is because if you think about the Harry Potter cloak, you think about Harry Potter wearing it and walking out in the sun, right? But the sunlight is not just one wavelength. It's a combination of a lot of wavelengths. So the problem with cloaking is that the refractive index of materials usually depend on the wavelength. Okay? So you can design, let's say, a cloak for red light, but if you use green light, your refractive index will be different. So that means that light may bend in a different way. So you may be able to design a cloak for Harry Potter for one color, but if he's walking out in the sun, because it has all the colors, it will not work. Okay? And one example that you see in everyday life, or an outcome of this, is actually when you shine white light in a prism, and if you look at the other side, you will see that it's a rainbow, and that's really the reason why. Because all these colors, in vacuum, they have the same, they feel the same refractive index, but as it goes inside the prism, it feels different refractive index and they bend by different amount. So when they come out, actually the white color, it kind of splits into all the different colors. Okay? So does that mean this is the end of the world, that we cannot do anything about cloaking? Well, not really. Actually, you can design cloaks based on specific requirements. For example, here, people design what's known as the carpet cloak. So I'll go over them one by one. So what they did first is that they shine light from top. So this is a flat surface. By flat surface, what it means is it's something that reflects light. You can think about it as a mirror. Okay? So they shine light and then they look at the reflected light here. Okay? Now next what they do is they put an object and then they again look at the reflected light. Now when they first look at the reflected light, they do it for 3 different colors. So that's blue, green and red, and they look at the reflection, how it looks. Now when they put that object, of course, because there's an object, your reflection changes, and that's why you see that now instead of 1 spot you have 2 spots, and here you have 2, and here for red light you have 3. Now they design a cloak, which they call as a carpet cloak. They put on top of it, and then you can see that this pattern looks very similar to what it was initially. Now what that means, it means like if you have a mirror here, light is shining from the top, so you're looking from the top, it looks like a mirror. Okay. Now on the mirror you put an object, you can see the object. Now if you put this cloak, now you cannot see the object anymore. What you see is again a mirror. So it is somehow making things invisible, but it's not really the invisibility cloak that you think about Harry Potter. So now just to summarize what I talked about, I showed you in principle that invisibility is possible. To make things invisible, you need to bend light in a very special way. And for that, you need to go beyond nature and you have to take help of what are known as the metamaterials. And finally, you may not be able to make a cloak for all wavelengths, but based on specific requirements or specific colors, you can actually design a cloak with this kind of principle. So now that I have told you that it's not that easy to make a Harry Potter cloak, that means people can see us, right? So we need to stay young and healthy. So now Lauren is going to teach us how to stay young, healthy, and beautiful.
So my name is Lauren, and today I'm going to talk to you about a few studies that were aimed at understanding what happened to us as we age and how we can prevent many of the detrimental things that happen to us while we're aging. So we like to refer to these studies as the scientist's guide to the elixir of life. So the quest for the elixir of life seemingly has existed forever and still exists in our daily lives. So hopefully after the last talk, we all know about Harry Potter and the Philosopher's Stone or the Sorcerer's Stone, where the user is able to live forever. And we know about this historical search for the Fountain of Youth. And we see anti-aging products all the time, geared at selling us this idea that it can prevent aging. And so it appears that as long as humans have existed, they've been sort of baffled by the causes of aging and how to prevent it. And this makes sense because not only is it an exciting idea, but it can also help to aid society by preventing aging. And this is because the National Institute of Aging predicts that by the year 2050, there'll be over 1.5 billion people over the age of 65. And it turns out that caring for disability and disease in aged populations is very expensive. So the longer that we can keep people healthy, the lower these costs will be. However, we also recognize that as our lifespan increases, our so-called health span, or the amount of time that we're alive and healthy, will decrease. And this begs the question, do we really want to increase our lifespan? But luckily for us, the goal of current research is more about increasing our health span. And so it's geared towards preventing and treating age-related diseases so that we can live healthier for longer. And there certainly is evidence that there are people who have a long lifespan and health span. For example, this is Emma Morano, who at 115 years old is the 5th oldest person in the world and the oldest person in Europe. And she recently had a birthday, and the New York Times asked her how she explained her long lifespan and health span. And she said that it's because she eats 3 raw eggs a day and has remained single since 1938. And while this is pretty entertaining, I think that scientists might explain it a little bit differently. So let's take a look at that. When we look at aging, we have to start on a very small level. Every part of our body is made up of these tiny cells. And these cells work together in order to form tissues that carry out functions in our body and eventually make up our organs, such as our heart. As we age, our cells accumulate damage, and this builds up in our tissues and eventually causes our organs to become dysfunctional. So if we want to understand the root of aging, we really have to move back to this cellular level. However, our bodies do have a way of repairing damage that occurs during our lifetime. We can probably all think of a time when we injured a tissue and it repaired itself, such as getting a cut on your skin that eventually will heal. And this is because we have special cells in our body called adult stem cells, whose job it is to repair our damaged tissues. So these are a little bit different than embryonic stem cells, which more people might be familiar with. These cells actually reside in all of our tissues, and what they do is divide indefinitely in order to form more of a specific cell type to repair any damage. So for example, we have cardiac stem cells in our heart, and when our heart is damaged, it can divide to generate more heart cells and repair that damage. However, as we age, this function is not as efficient. So when we do get damage in our cells, this will remain and build up in our organs. For this reason, since all of our cells decline in function as we age, this means that all of our organs also decline in function as we age. So we can see that aging is really systemic and affects all parts of the body, everything from our brain to our heart and lungs, and even we get gray hair and wrinkly skin. So a few labs took note of these specific things that we know about aging, and they wanted to research how we might be able to prevent this decline in function of our cells and our organs. So they knew that aging is partially a result of our declining adult stem cell function, and that this results in a declining tissue function, and that this occurs everywhere in the body. So they thought about these different points, and they thought that in order to affect all of this adult stem cells in our body, there might be something that prevents aging that's actually circulating throughout our whole system. And what do we know that circulates throughout the entire body? Our bloodstream. So that is exactly where they started looking, for rejuvenating factors in our bloodstream. To study this, they had to take, a very peculiar approach where they actually joined the circulatory system of aged and young mice in a method that's called parabiosis. So you can think of this as sort of generating your own conjoined twins. And this way, anything that's in the blood of the young mice will now be exposed to the aged mice as well. So if there's any rejuvenating factors present, now the aged mice might experience reversal of aging. And as controls, they also looked at young mice alone and old mice alone, which should show no difference. Their first experiment was looking specifically at the function of the adult stem cells in repairing tissue damage that occurs in aged mice. So they performed parabiosis, and then they actually damaged the muscle tissue using dry ice, which generates a very distinct border between the healthy and the damaged tissue. Then they waited 5 days and looked to see if the adult stem cells had repaired any of this tissue. You can see that in the aged mice, there was not very much repair, and this damaged area still exists. In the young mice, you can hardly tell where the damage even occurred because it had repaired so well. And what was really shocking is that after they performed parabiosis, this aged mouse had an enhanced ability to regenerate its muscle tissue after damage, almost as well as the young mouse in the pair. So to summarize what this experiment tells us, they found that there does appear to be some kind of rejuvenation factor located in the bloodstream of young mice. And this circulates in the young mice, but is absent in the aged mice. That way, when they perform parabiosis, this particle from the young mouse now is circulating through the aged mouse, and they get the benefits as well. So if we go back to these adult stem cells that are responsible for repairing this tissue damage, and that typically do not function well in aged mice, this means that when they're exposed to this rejuvenating factor from the young mouse, all of a sudden they're better able to function and repair these tissues. So by now they'd established that parabiosis helped to repair damage more efficiently, but now what they wanted to look at was to see if actually the blood from young mice could prevent or even reverse aging. And one great example that they looked at was the heart. So it's known that the heart actually enlarges and has thickened walls in aged mice. And this is really important because this happens in humans as well. And this is what causes some of the dysfunction as we age. So what you can tell is that when they looked at young mice, it appears to be a very normal sized tissue. And when they looked at the aged mice, they saw that it was very, very large and probably dysfunctional. When they performed parabiosis, they saw hardly any change in the young mouse, but they saw a drastic size reduction in the aged mouse. So they found that it actually had gotten smaller, so these rejuvenating factors were not only able to repair damage, but they were also able to reverse damage that had already accumulated. So basically, we know that there seems to be something in young blood that can help us live forever. So it seems like we already know the answer to this whole immortality thing, and we just have to become vampires. So it seems like they really had this figured out a long time ago because they drink blood and they live forever. So it's a really great solution, except it's really gross. So luckily for us, it doesn't have to be that complicated or gross, because maybe we don't need to be exposed to everything in the blood. Maybe all we need is a few small factors. And that would be enough to prevent and repair the function of our aging organs. So to find out if there's only one or a few small factors that we need, a very simple experiment was performed where the blood was compared between young mice and aged mice, and they looked to see what was different between the two samples. One particular protein, called GDF11, stood out because it was present at very high levels in the young mice, but very low levels in the aged mice. Then, after they performed parabiosis, they saw that the aged mouse actually had fully restored levels of GDF11 protein. And it turns out we don't know much about this GDF11 protein, but what we do know is really important for aging, and that is that it acts on these adult stem cells in order to enhance their function and help them be able to regenerate tissues. So next they wanted to see if this protein on its own was able to cause these similar reversing of aging signs. So they did this either by taking just the protein and adding it to the blood of aged mice, or taking the plasma from young mice, which contains all of those proteins in the blood, and adding it to aged mice. So now there's no need for any parabiosis or joining any circulatory systems. And they found that this alone was able to act as a rejuvenating factor for many different organs, including our heart, brain, and even our sense of smell. So what does this mean for humans? Can we all just start taking huge amounts of GDF-11 and be healthy forever? Unfortunately, it's not that easy because there's still a lot of things that we don't understand about GDF-11. We actually don't know how it works, and we don't know if human and mice act the same. So just because it works in mice doesn't mean that it's going to work in humans. We also don't know why GDF-11 decreases naturally in our bloodstream as we age. Maybe there's a reason it's very low, and if we increase it, it will actually be harmful. As far as treatments go, we don't know the side effects, what age to start taking it, what dose to use, or how long to take it for, which are all very difficult questions. We also don't know if it's safe or if it even has an effect on the lifespan, because that was never actually measured in the mice. Luckily, there are studies going on in both mice and humans to help address some of these questions. And specifically for humans, there is a clinical trial started just this year at Stanford University called the Plasma for Alzheimer's Symptom Amelioration, or PLASMA study. And in this study, Alzheimer's patients are given plasma from healthy 30-year-olds to learn more about the possibility of using this as a treatment to restore the cognitive function in Alzheimer's patients. So since this has just started, right now all they want to do is establish if this is a safe procedure to use. But you can imagine that if they do find a specific protocol, future studies will look to see if the cognitive function of Alzheimer's patients actually increases after exposure. The great news is that if we can answer all of these questions and figure out a treatment plan for various age-related diseases, it's actually really feasible. First of all, this is something that's already in our blood, so we know that our body can tolerate it. And additionally, it can be introduced by a simple blood or plasma transfusion, which happens all the time in hospitals for various reasons. The only problem is that likely this will require such a large amount of blood or plasma, and eventually it would be useful to find a way to create a GDF protein pill or combine it with other anti-aging factors that we discover. So in the future, we might not be able to live forever, but we are definitely well on our way to living healthier for longer. Further research into GDF-11 and other anti-aging factors will continue to make this a reality. So now that we have found ways to help prevent us from aging, but what happens if we're already dead? So now we need to figure out how to bring things back from the dead, and I'm going to turn that over to Matt.
Hey Norm, my name is Matt, and I'll be talking about raising the dead. Can we actually clone something like a woolly mammoth? So there is an absolutely amazing diversity of life here on Earth. But we humans are actually doing a really good job of driving many of these species extinct through pollution, overhunting, and habitat loss. In fact, about 4 species go extinct every hour. And often when one species goes extinct, it causes a cascade effect that affects the entire ecosystem. Take the tiny island of Mauritius in the Indian Ocean, for example. On Mauritius, there used to live a giant tortoise, but when humans arrived, this tortoise started to go extinct. And this was important because the tortoise ate the fruit of the Mauritian ebony tree. And so when the tortoise went extinct, these fruit were not being eaten, the seeds were not being dispersed anymore, and the Mauritian ebony tree started to go extinct. And humans were concerned when the ebony tree started to go extinct. Not only could we lose all the species that were living in this tree or that fed on this tree, but Mauritians lose ebony wood, which they use to make musical instruments. And without the ebony tree, we can't make things like black piano keys. And so, if we can't make things like black piano keys, the economy of Mauritius collapses. I mean, this could have been even worse, right? What if this tree had some kind of life-saving medicine that we all needed? The point is that everything is connected, and often extinctions can have downstream effects that we don't really anticipate. But what if we could reverse these cascades of extinction? What if we could bring species like the Mauritian giant tortoise back to life? What if we could actually clone extinct species? I know what you're thinking, science fiction. Actually, no, this is science fact. The Pyrenean ibex went extinct in 2000, and it was the first species that went extinct to be cloned in just 2003. We've also cloned the gastric brooding frog in 2013. This is a species that's so weird it would eat its own eggs and then hatch its young inside its belly. Now I know what you're thinking, sweet, we can make Jurassic Park happen, right? Well, actually we can't. That's still science fiction. And why is that? We need a little basic review in biology. So as Lauren said, our bodies are all made up of tiny cells. And inside the cell, we have something called the nucleus that acts as the control center for our cell. And inside the nucleus, we have strands of DNA. This DNA is made up of 4 chemicals we call nucleotides. And these nucleotides form our genetic code. And this is basically the blueprint for all living things. It's what makes us, us. This is just a small piece of what our genetic code as humans looks like. We have over 3.2 billion of these letters in our genetic code. So to clone anything, we need this blueprint. We need DNA. So could we clone T-Rex? Can we actually find T-Rex DNA? Well, T-Rex lived about 65 million years ago in the Cretaceous period. And here we are now, at 0 million years ago. What's the oldest DNA we've ever found? Only about 700,000 years. Way short. In fact, we think that DNA can probably only survive for about 1 million years, because as soon as you die, your DNA starts breaking down. But 700,000 years is still a really long time, right? How did DNA even last that long? Well, this DNA came from a Pleistocene horse that was frozen solid in permafrost for that entire time. Freezing is a really good way to preserve DNA. So if we want to clone any extinct species, they have to have died and gone extinct within the past 700,000 years. So what lived during the past 700,000 years? Lots of really cool things. For example, the dire wolf. Yes, a real animal that lived here in North America. The saber-tooth. The moa, a giant bird from New Zealand. The passenger pigeon, an icon of extinction from right here in America. And the Tasmanian tiger, a marsupial predator from Australia. And most importantly of all, the woolly mammoth. Woolly mammoths were giant hairy beasts, about the same size as today's elephants, but a little bit heavier. And they lived during the Pleistocene epoch, also known as the Ice Age, when glaciers covered much of the northern hemisphere. And our ancestors have been fascinated with mammoths for a long time. We painted pictures of them, we hunted them for food and clothing. We even built our houses out of their bones. And even today, people are still hunting mammoths, digging up their ivory so they can actually carve it. The mammoth would be the ultimate poster child for cloning extinct species. If we could clone the mammoth, we could raise awareness and start cloning many other species. And actually, it's a pretty good one to start with. Remember how I said that freezing things was a good way to preserve DNA? Well, mammoths lived and died where it was very cold, so we can get their DNA. And mammoths are the species that people are most interested in bringing back to life. Every time I give a talk or go to the museum, someone asks me, when are we going to clone the mammoth? So, before we learn how to clone something that's extinct, we have to know how to clone something that is still alive. Does anybody remember the first mammal that we cloned? Dolly. That's right. Dolly the sheep, cloned in 1996. So how did we actually do that? Well, first we take the animal we want to clone, and then we take out a skin cell or some other cell of it, and then we suck the nucleus out of this cell. Now, why would we want to do that? We suck the nucleus out because we want the DNA of this animal. But we don't actually want that cell, because this cell is stuck as whatever kind of adult cell we pulled out. If we pulled out a skin cell, this cell could only multiply again to form another skin cell. And if we're going to clone an animal, we need brain cells and heart cells and hair cells and everything else. So we need something that can transform into any cell in the body. But we need our embryonic stem cells. And we get those from an egg donor. So we take our egg donor, we take out an egg, but we don't want this DNA, right? Because it's the wrong DNA. So we suck that DNA out, and we put in the nucleus with the DNA that we actually wanted. So now we've got a cell with the right DNA that can multiply and form into any type of cell in our body. But how do we get it to start dividing? Well, we zap it with a little bit of electricity and we go, "It's alive!" That's actually what we do. We zap it with some electricity, and that gets the cell starting to divide into an embryo. And once we get that embryo, we place it in a surrogate mother. Then we wait, and in a little while, a clone baby is born. Now this clone doesn't look anything like its mother, right? Because that's not where it's got its DNA. It's got its DNA from the original sheep that we wanted to clone. So since we've cloned Dolly in 1996, we've gotten pretty good at this. We've cloned all sorts of mammals. And we've gotten better at making our clones too. For example, in 2013, this cow named Apple won the second place at the World Dairy Championships. Who beat out Apple? Apple's own clone. Also in that same year, this cloned horse helped win one of the largest polo matches in the world. And you too can get in on the action for just a paltry sum of money. You could have your favorite furry friend cloned to live on forever. So it's all these recent advances in cloning that make people think maybe we can actually start cloning the woolly mammoth. But how would we do that? Well, pretty much the same way, right? We'd want to take a cell from the mammoth that we want to clone. And it seems like that would be pretty easy because we find frozen carcasses of mammoths all the time. The problem is when we look at these cells, they're too damaged to use. They don't have good DNA. But thankfully we thought of a way around that. Woolly mammoths are actually pretty closely related to Asian elephants. They may have only diverged about 3 million years ago. So, if we find mammoth DNA, we can put that mammoth DNA into an elephant nucleus, and we can use an Asian elephant as a surrogate mother for our mammoth. So, where do we find good mammoth DNA? Surprisingly not in really good mammoth bodies. For reasons we don't quite understand, well-preserved bodies don't preserve good DNA. But we can get good DNA just out of skeletons, which don't have well-preserved bodies. So, we cut open the bones, we suck out the DNA, and this is what elephant DNA would look like. Good, fresh, modern DNA. But when we look at ancient DNA, it's all broken and up, not in order. We don't know where everything goes. We're missing pieces. But remember, elephants and mammoths are pretty closely related. They probably have about 98% of the same DNA. So we can use the elephant DNA as a guide to arrange our mammoth DNA. This helps us put the mammoth DNA in order. But not all of the DNA will actually match up, right? So here in red, this is where our genetic code for our mammoth is different. And that's okay. We actually want that. Because where it's different from an elephant, this is the DNA that makes mammoths mammoths, and not elephants. So we have our mammoth DNA, we're still missing big chunks, right? Because it's all broken up. We could fill in those chunks, but what's actually easier is just to take our mammoth DNA and splice it in to our elephant DNA, where it's different. In real life, we'd probably have to make about 70 million of these changes spliced into elephant DNA to make our elephant DNA into mammoth. It sounds really complicated, but this is actually the easiest way to get a full strands of mostly mammoth DNA. So once we have that, we can take an adult cell from an elephant, we splice in our woolly mammoth DNA, we get it to dividing into an embryo, we put that embryo into an elephant, and then we wait almost 2 years. That's how long it takes for an elephant to have a baby. And then our clone mammoth baby is born. But remember, our DNA was actually a hybrid, a mix of mammoth and elephant. So all these places where it's gray, that's elephant DNA. So really what we get is a hybrid baby. That's part mammoth and part elephant. But even if we had 100% mammoth baby, would it still grow up to be 100% a mammoth? Probably not. Because it's not going to learn how to be a mammoth, right? Who's going to be its mammoth dad and teach it how to do mammoth things? No one. It's going to learn how to be an elephant. DNA isn't everything. And we know this because of studies we've done on human clones. Yes, that's right. Human clones. They walk among us as identical twins. Identical twins are actually genetic clones of each other. And they may look alike and they may act alike, but they're not the same person, right? And if twins make different choices and live different lives, they're going to look different. For example, this twin on the right likes to smoke and sit out in the sun, and her skin shows it. So let's say we get everything right, and we finally get our 100% mammoth. That's good for a zoo, but that doesn't really bring the species back, right? We're going to want a whole herd of mammoths, a whole population out there, and that's going to require a lot more cloning. And once we get this herd of mammoths, can we make sure they don't go extinct again? I mean, if we put them out there, are they going to have a place to live? All these species that I talked about before, we killed them, right? If we brought these back to life by cloning them, what would stop us from just killing them again? So, how close are we to actually cloning a woolly mammoth? So first, we need to sequence the mammoth DNA. And we're actually doing really well on that. In just 1 or 2 years, we'll probably have every single bit of the genetic code of a mammoth. Then we need to splice that mammoth DNA into elephant DNA. And actually, just a couple weeks ago, a professor at Harvard named George Church said he had done this. Now, he didn't make all 70 million changes, but he showed that it was possible. And actually, this last one, finding a place for the mammoth to live, we're doing really well on that too. There's actually several parks in Europe that are set up, ready to go for when the mammoth comes. They have the right kind of grasses, they have animals that would have lived there at the same time as the mammoth, so they're ready. It's the step where we actually birth a baby that we haven't tried yet. And that's something we have to be a little bit cautious about. Because cloning is a complicated process, and it doesn't always work. So for example, when they made Dolly, they needed 277 of these embryos just to get one healthy baby out the end. When they cloned Snuppy, the first dog, they needed over 1,000 embryos and hundreds of surrogate mothers just to make one healthy dog. We don't actually have a lot of elephants to use for this, right? Elephants are endangered. There may be only 40,000 left. We can't take a huge part of that population just to make mammoth babies. We need elephants to make elephant babies. And cloning extinct species is even harder than cloning species that are still alive. For example, the Peridian ibex I talked about, yes, we cloned it, the baby was born, but it died just a couple minutes later. It had horribly mutated lungs. And the gastric brooding frog, we've only got this to embryo stage. It hasn't grown all the way into an adult frog yet. And furthermore, elephants are really smart, and they're emotional. They have feelings, right? What's an elephant mother going to think when its baby comes out looking like this? Is it going to love that baby? Is the baby going to grow up in the herd and be accepted? Will he be ostracized? Do we want a really sad mammoth baby walking around that doesn't have anyone to love him? And you'd say, well, come on, it's only a 2% difference in DNA. But I would remind you that there's really only a 2% difference between us and our next of kin, our hairier relatives, the chimpanzees. How would you like it if someone told you to have a chimpanzee baby? So, these things that we've already started doing, we should continue to do those. It's good basic research. It teaches us about mammoths. This can actually be used for health. We can splice out cancer genes in our own body. And these parks are good too. They help us raise land for animals that we might want to preserve. But I think we're going to have to be really careful about this step right here. And we should probably hold off on this. Let's focus on saving elephants before they go extinct so we don't have to clone them afterwards. It's a lot easier to save something before it's dead. So, to sum up, Jurassic Park, totally fiction. But Pleistocene Park with woolly mammoths, totally possible, theoretically, but I don't think we should do it. So thank you.